Thank you for coming today, and thank you for giving me this opportunity to just tell you a little bit more about what we do at the Protein Expression Facility to support research across a huge range of fields. To be politically correct, actually, we are a central facility supported by UQ and also a few institute and faculty, so QAFI is one of them. So we are physically located at AIBN, but we actually are not in AIBNs. So just want to make sure that's correct, or else I get chopped. All right, so today the aim is really just to give you an overview about like making proteins, and a lot of you will actually be making proteins for whatever applications that you need, and a lot of you will probably face common challenges. So here what I want to do is tell you some of the strategy you probably can employ to overcome, and some of the technologies that we offer, which we're happy to work with you in close partnership. And we are working with a lot of academic group, industry, as well as some government organization. It's a challenge, but sometimes it's a good challenge. So I'll tell you about our capabilities, what we have here. So we enable world-class research, and up there, as I say, across discovery and translational, very diverse research needs, in say diagnostic purpose or reagent generation for assay, crystallography, drug discovery, screening, also in agriculture science like vaccine for animal and also human vaccine. So we've worked with actually more than 100 research groups at UQ, and we understand the I guess the demands of different research needs. We also provide what we call upskill professional development. So you might have research staff that you want them to be trained in a particular skill in the area of protein production. We actually do conduct that training, and they can then access the facility to say produce the protein that you want. We also do tailor workshops so we've been invited to do workshops in other countries where we go in, we do either give a series of talks or web labs to teach the students or the research staff all the different processes about protein production. So I just wanted to give you a quick overview of say what we do in terms of our pipeline, and so we do anything from cloning all the way through to protein purification. And as a user, you can enter at any point you like, and you can leave the pipeline at any point you like, and I'll tell you a little bit more. So the first thing is, of course, get your expression construct. So we can do that if you only have a few constructs, a customized one, or if you have a lot of constructs. Some people come to us with 48 constructs or 96 constructs. So we do it in a high throughput format. And then we take them through expression screening. So we are actually the only facility in Australia that offers, expression in all these systems. So we have the bacteria, E. coli, as most people would know, yeast, Saccharomyces cerevisiae, Pichia, K. lactis, insect cell baculovirus, mammalian, and we also offer a cell-free, which is a eukaryotic cell-free system that's made in-house. After we do the screening, we find out what's the best system to use, what's the best condition. Then of course you have to scale it up. But sometimes people prefer to scale down. So for example, if you're looking at say the functions of a huge range of gene, or you want to look at if you change the messenger RNA codons of a particular gene in hundreds different ways, what sort of output do you get? So we can actually do scale down to where we do it in 24 deep well plates, so you don't actually have to scale up. But it can go both ways, whether it's scale down or scale up. So for scale up, we can do it in shake flask all the way through to bioreactor, where it's up to 20 liters bioreactor fermentation in all the systems. And then once you of course express your protein, you need to purify it, and we purify proteins from intracellular, extracellular fractions. And then we use a sort of different chromatographic techniques to do that. And at the end of it, I guess for, say, the animal group in QAFI, like you guys want to do it as develop vaccines, we'll make sure that it's endotoxin free before you actually inject it into the animal. So that just gives you an idea, of what we have. So we have a very robust and flexible pipeline. And it's actually quite complex because each steps you have so many different options. And we are set up and we have been established to have all these different vectors, all these different systems, and all the possible combination you can think of in the facility. And the aim is really to serve and to enable research at UQ. So, the advantage is, we can do a whole combination to try to get you the proteins of interest and what you really need. One common question that people tend to ask me, or the first question they ask me is, I have this protein, tell me what system I can produce it in and tell me how much I get. And I can't give you the answer because I wish there's a universal system that can produce all proteins in all the quantities that you ever wanted, because if I know that system, I'll hit the lottery and the jackpot. But I don't. I'm still working, so I haven't found the answer yet. You can see that actually the different systems, so you have mammalian, bacterial insect cells, yeast, bacteria, and I was talking about cell-free. And cell-free can be E. coli based, wheat germ based, or we have the Leishmania, which is eukaryotic based. So you can see that, depending on your criteria, it varies. There are pros and cons of each system. There's benefits of using each system. So therefore, the importance is really knowing what you're trying to produce and what you want to use it for. So this is an example of, say, most of the time it's like a student coming to us and say, okay, tell me which expression, to use. And then they say, when we try to tell them that each expression system performs slightly different. So your post-translational modification is different. So for example here, this shows you, the different glycosylation you're going to get on your proteins, depending on what system you use. And most of the time they're like, okay, this is too hard. I just want protein. Don't tell me all these stories. Don't tell me all this information. It's just too hard. I just want protein. Tell me which system to use. And at the end of it, they say, really, I just want proteins. But what we need to tell them is, you might get that tube, of solution. But what you really need to know is it denatured, aggregated proteins? Is it a monomer, or is it, oligomers, or does it actually assemble in some sort of protein complexes? So for example, virus-like particle, which would be, the quaternary structure. So it's really important to know all that, because only when you have actually analyze, characterize your protein, then you can take it onto your application. So for your application, it could be very varied. Some people want to know the structure. Some people want to know the function. I found a novel peptide. I found a novel protein. Would it do this? I speculate, or my hypothesis is it would do this function. I want to prove it. So only when we know that we fully characterize your protein, then we can have the confidence to say when you do your application, the result you get, you can trust it. So commonly, the thing is, okay, I've cloned it into a vector. Would it express or would it not express? In the best case scenario, which is what I'm going to show you, is you clone it, it's successful, it expressed, great. You purify it, you got your protein of interest. Some people put a tag on it. You remove the tag, it's still soluble, it stays in solution, you're happy, you characterize it. It's great, it's perfect. It's a perfect world. But it doesn't always happen this way. From my experience working with hundreds of protein, I would say that usually you have to do a lot more hard work for it to go through this so smoothly. And it's really important to understand each stage that you're doing this. So when it's not so ideal, say, you start with expression and you go, okay, is it expressed? Is it not expressed? So when you don't get expression, you ask yourself, what's next? What should I do? And then if, say, you do get expression, it's all insoluble. As you can see here, this is insoluble fraction. Say this is soluble fraction. The proteins are all in inclusion body, and you can't use them because they are not folded correctly. So they don't have the biological activity that you want. So some proteins, you can use it as a denatured form. But a lot of proteins, people want it in its right tertiary structure. It can't be denatured. So what's next, And then when you try to purify it, you go like, okay, I'm just getting all this, co-purified host protein. Is not really purified. So what do I do? So my aim today is, to cover some common strategy and also to highlight some common pitfalls that people tend to have when they do it in the lab, and hopefully that will help. And then actually from the group leader or the principal investigator point of view, is generally like, time is really important. These days, research, we're generally pushing outcomes all the time. So time is important, getting the right quality, keeping the cost really low. All these are important factors that we have to consider. So that's one of the reasons why UQ is supporting us, to try to serve research here. So to start off with, I want to give you a number of case studies to talk you through it, at the same time highlight the strategies that you can take and some of the technologies we offer. But to start, you really need to know your protein. I know it sounds simple, but a lot of times when I talk to new users, the first thing they say to me is, oh, I just found this sequence, I don't know what it does. But it's really important, and that's what we do. We do a lot of search into your protein to understand what's the protein before we decide which way or what's the best strategy we should take. Because your protein could be glycosylated, then we can't use E. coli, because it needs the glycosylation for it to be stable, for it to be biologically active. Or your protein, might actually needs to be secreted, so we shouldn't express it as intracellular. It depends on the protein, and it's really important for us to know your protein before we move forward. And the other thing is, this is one thing I keep saying to a lot of, especially PhD students, you start well, you really will end well. It's very important. A lot of times, and we see this across all the UQ research lab, is they usually go, oh, I'll go next door and see what vectors they have, and I'll just use their vectors because I don't have to buy vectors. They have a plate, I could just do some, and then all is good. But if you don't start well in terms of considering some of the factors, when you come to, say, expressing it and purifying it, you might find that it's in the wrong vector, it doesn't have the right feature, you have to start again. So we always tell people it's very important to know what you want. So your choice of expression vectors to go into each system is really, really important. So that's what I mean by, is vector choice important? Yes, of course it's important, because every system, you need different regulatory elements, to control the expression or to promote the expression. And each expression system has slightly different, regulatory elements. So it's important to know that. And some proteins, you might need a fusion protein or solubility tag, on the N terminal. So you want to pick the right one. And also, if your protein, say it's a mammalian protein, but you want to produce a domain of it in E. coli, you know that it will be soluble, but then you want to consider whether does it have red codons. Should you optimize the genes from the start rather than later? Because if you go through the cloning, the expression screening, you find that it's insoluble, you've done everything you can, it's still insoluble, then you might have to go all the way back to say, okay, let me codon optimize it and find out, okay, now it's soluble. So it's better to actually look into those parameters at the start than at the end. So I will run through some of it. The other thing is also maximizing resources. We know that these days, research grants get smaller and smaller. Generally, it gets cut. And it's really important, how we can maximize it. So previously would be you have one expression vector, it goes into one expression system. But actually this day, we are able to offer a vector that allows you to go into 3 expression systems at the same time in parallel, which means that you don't have to reclone. So you clone it once and you say, I would like you to test the expression in these 3 hosts at the same time. Or test one first. If that didn't work, then move to the next one. But we don't actually have to reclone. That would save time and resources. So that's one thing to consider. And we've tested this vector. It works well. The other thing is time. So some vectors, it will affect your downstream in terms of what else you need to do. And you can see that for here, an example I point out is this particular system, you are able to get your first result in 10 to 15 days, okay? But the other system, which is really popular and cheaper, but actually it takes longer to do. So time is actually really valuable, and time is money, which I've been trying to stress to students too. And sometimes it's better to use the right system from the start, so that we don't actually waste any time doing it. So some case studies. So in this one, I will point out in terms of E. coli how to take insoluble proteins. What are the things that you can do in the lab to try to bring it to be soluble protein. So remember, I was showing you this slide about what if it's not expressed or what if it's insoluble, and talking about overcoming the challenge of getting soluble protein. So for E. coli, it's really important for people to note that these three factors are really important. So the vector that you choose, the host strains, and the culture conditions, and they actually work together. So you can't say, I only want to test host strains and forget about culture condition, or I want to test different vectors, but I don't want to test the host strain. It's actually a combination of it that's really important. And to do that, that means that you have to do screening. And then people would generally ask, like, how many do I have to screen? How many samples do I have to collect? So what we do, in general, for any proteins that comes in E. coli, in a single vector, we'll put it into 2 host strains, we put it into 2 media, and put it into 2 temperature. So at the end of it, you can see that you do end up to close to anything between 25 to 50 samples, depending on what samples you want to analyze. And so it's actually like another question that people ask is, if I have to screen that many, how many flasks do I have? What do I need to do? It's such a mess having all these flasks. And we see that very often actually in some labs. So what we want to tell you is you can actually improve the throughput. That would be doing it instead of shake flasks, do it in 24 deep well plate. Go the scale down method. And we do this routinely all the time. And we have tested it and show that it is just as good in shake flasks. So we do our cloning in 1 to 2 weeks, so that if you have a lot of construct, that can be done in a 96 well plate. And then we usually take all that into 24 deep well plate expression screening. For E. coli, that takes about 2 to 3 days. And at the end of it, if you really have a lot of constructs, then we have to run the whole analysis. And we do have a lab chip that allows us to run 384 samples at one time to analyze the result. So you don't always have to do it in shake flasks. That's one thing to remember. It's actually better for you to screen more conditions and do the scale down. So host strains and temperature effects. So this just gives you an example. So this is a membrane-associated complex. So initially, the user put it into a strain called Rosetta, and they say, we don't get expression. So then they pass it over to us. So what we did was, then we look at different, combination, what we have to do. So generally, like I say, we do look at temperature. So this is in the same host strain in temperature. You see, we still can't see the expression, but when we switch the host strains, you do then get expression. But unfortunately, this is an insoluble. So no matter what we do, even though we've done a lot of screening, it remains in the insoluble fraction. Okay, so you don't always get everything that you want, but it's actually really important to screen strains and temperature. So another much more positive one would be, this is a plant pathogen response protein. So initially when done at 37 degrees, as you can see, it's insoluble. You don't get any soluble protein expression. So we actually did look at different strains and also change the temperature, and then you start getting your protein that you want. And from there, they were able to use it for their assay and then publish from there. So that's the importance of strains and temperature. In E. coli expression, temperature is actually really, really important. The other thing about overcoming the challenge is, yes, you got your expression is soluble. The next thing is actually trying to purify it. Purification, protein recovery and protein purification is not minor. It actually requires a lot of time and understanding the process and the protein. So this is to show you, so you have a case where, say, this is the elution from the first step of purification. You will look at me and go, okay, this is your protein of interest. Is that purification? You still have all this band. Yes, that's true. So this particular protein co-purify with the host protein, and it likes to do that. So the exactly same process for different proteins works really well. But for this particular protein, it was a lot more difficult. It likes to co-purify, and that's what we get. And the other thing we realized that when we store it at 4 degrees overnight, it starts to precipitate out. So you start losing your protein of interest, which is what you see. So what we need to do is, take it from here and to here, which is at the end, you only want a single band, and you want it to be stable after you store it. You want minimum loss of proteins. And how do we do that? I don't expect you to actually look at all that. I just want to show you that we do also plate format screening of buffers to help us to look at how to improve the stability of proteins. So we actually look at, say, what's the optimal purification buffer to enable the binding of your protein of interest, but not the binding of, say, the whole cell protein. And we also look at, the different buffers and additives that's needed to actually keep your protein stable in solution, or when you freeze store it, it doesn't precipitate out. So all this can be done in a plate format, and we do that to try to get you the optimal condition. So at the end of it, then you can actually say, okay, this is what I have and I can use it now. So it's actually also really important and really useful to do that screen when you do purification. So for E. coli express protein, I just want to highlight this, that it's actually really important for you to remove the endotoxin after you produce the protein. So endotoxin comes from the bacteria cell wall, and a lot of times you will cold purify it, and endotoxin is also found on, say, a lot of the beakers or borders that you use if it's not cleaned properly. So we have a very sensitive technique that allows us to measure the endotoxin level really well, and this is an FDA approved method. Also at the same time, we have used different techniques to remove endotoxin. So for people who are developing vaccines to put into animal, this is one thing to consider. So another case study is actually, say, changing systems. So we talk about E. coli. We're now going to talk about, say, insect cells and, producing secreted glycoproteins. This is a class of proteins that, actually a lot of people are interested in or use for different assays. And insect cell is actually a really popular system for producing recombinant proteins. So some of the common challenges people have when they produce glycoproteins, so is low expression. So for example, if you look at this gel image, this is the cell palette. You have it all in the cell palette, but you can't see it in your supernatant, which is useless because you want it to be secreted. When you do a Western, it's really low, and that's just not enough for what you want to do. You have to deal with very large volume of supernatant, so what do you do to try to recover that protein? The other thing is the poor target protein recovery and purity is another issue when you try to purify from insect cell supernatant, because there's actually a lot of proteins that likes to bind to, say, the nickel column. And then you also have contaminating proteins you try to separate out. So I'll go through, what do we do in our facility to overcome some of this issue, or, when projects comes in as such, how do we, deal with it? So for glycoproteins, it's really important for you to try to secrete it into the supernatant, which means the signal peptide. The choice of signal peptide is important. A lot of vectors comes with certain signal peptide, and it's common for most people to say, oh, let's just pick the vector because it's in the lab and it has that signal peptide. But it might not be the ideal one. So we actually do have a range of signal peptide that we can use. We have also different baculovirus system, because the different system can give you slightly different yield and benefits. And the cell line, we screen the cell line. And the other thing is temperature. As I say, for E. coli, temperature is really important factor to get expression and soluble expression. It's the same here in insect cell. It can affect the stability of your protein and the expression. So this just shows you the two systems we have. So the back-to-back system, don't worry, it's just another system. The back-to-back system, it goes through the E. coli step, and then it actually takes longer, whereas another system we use actually is a lot faster. And there are differences between the two systems. Like I said earlier, we do all this in deep well plate now. So basically, if you come to us with such a project, we'll do the cloning. If it's one single one, then we wouldn't do it at 96 well plate. But if you have a lot, we'll do the cloning within 1 to 2 weeks, and then from there on, we do the recombination, which is the transfection recombination, to get your recombinant baculovirus expressing your protein of interest. From there on, then go into amplification in deep well plate, and then expression screening in deep well plate. So by the end of day 13, you should have your first result. And so we have really optimized this to do it in the shortest possible time. And so this is commonly done and done daily, in the lab. So from that project, as you can see, this is the full data set then you will get out of it, just from the 24 deep well plate. I don't expect you to go through it, because it's just, I just want to tell you how rich the data would be, and you can then sit down and actually analyze to see what's the effect of, say, temperature on that particular protein. Because in this case, I actually have 3 different proteins in there. So does the cell line affect my protein? Does the system affect my protein? What about temperature? What's the best time of harvest? So it's really important to do that screening to find that condition and the optimal parameters before you move on. One common issue we see in a lot of labs is when they passage baculovirus stock in the lab. They over passage it. So we usually make sure that we keep the passage number really low. So we do all expression at passage 5, but we do see a lot of group doing expression at passage 7, 8, 9, and 10. And this is commonly what happens in baculovirus. At passage 3, that's your protein of interest. You can see there's some truncation. So that's the expression, passage 4, 5, 6, 7, 8. And we've done further on, you see a drop. And what happens is you get more of the truncation. So this is an example of one protein, but I've seen this across so many proteins. There's only one single protein that I work with that's really stable across, which I was so surprised. I thought I've done something wrong. And I checked it again, and it was stable. So this is something that any students who's working with baculovirus, you really need to pay attention to, because even the industry, they were talking to us, they say, oh, I did the expression at passage 5, and then sometimes it worked, sometimes it didn't. I'm like, that's exactly what's happening, because, you just missed that one passage, the yield will drop off. Another thing about purifying, proteins from the supernatant is making sure you're using the right chromatography media. So a lot of people will produce their protein with a his tag, the 6-histidine tag, which is easy way to fish out your protein of interest. But if you don't use the right chromatographic media, what happens is you're actually losing your protein. So I'll show you here, for example, this is commonly used by most labs, and what happens is if you pass the supernatant through it, most of it will not bind. It will just go through the flow through. So this is exactly the same prep, okay, using 2 different type of chromatographic media. Whereas, using the newer Excel column, what happens is, you get a lot more protein of interest. As you can see, this is less than 0.1 mg total protein, and whereas this one, you get 8 mg. So that's a huge difference. And sometimes, it's really important to pay attention to such subtlety. So here I want to show you the effect of cell line on the protein that you express, and therefore what you need to do when you develop that purification protocol to recover your protein. So this is a primary purification step, IMAC, which is based on the his tag, so ion exchange and then size exclusion, so polishing step. So if you're same starting material but produced in different cell line, as you can see in high-5 cell line, which is commonly used to secrete protein because it's high expressor and it likes to secrete protein, but you do get a lot more background and contaminating protein compared to SF9 cell line. For SF9 cell line, you can get away with just the next step of ion exchange, but for high-5, you actually need to do another polishing step. The yield is higher for high-5 cell line. But what I want to show you next is, you really need to do the analysis. So I was quite surprised when I first did this years ago, as the same protein produced in 2 different cell lines and purified. What we looked at is, so this is high-5, it gives us mainly the monomer when we run it through the size exclusion chromatography analysis. Whereas when we actually produce the SF9 one, we realize that you do get the monomer, dimer, and tetramer. So of course the question is, which one is the fraction that we need to use? Which one is the functional one or the biologically active one? The monomer is useless. It's pure, but it's just useless to us. And then the dimer is the one that actually works and gives us activity. But the tetramer is really the one that is really good and useful. So then from there on, we learn to actually modify our expression and purification process to get mainly the tetramer that is useful for that project. So it's really important to go through and do this analysis to test, which fraction of the proteins is useful to you. Then assuming that if you pull everything together, it will be fine. So Just a few more. So one of it is antibody purification. I'm not sure whether anybody here does antibody purification. So we do produce antibody and purify it for people. Could be from hybridoma or from, say, HEK293 cells or CHO cells. And you go through, a series of steps to get really pure antibody. But please remember that you do then need to make sure you do the right analysis to check that your antibody is in its right quaternary structure and form, and you're not getting aggregates. A lot of times we do see like some people producing antibody and there's a huge aggregate peaks here, which means your protein is just all coming together and you won't be able to use it for your application. Another thing that we do work with others, and this is an industry project, is they produce virus-like particles. So this day a lot of people are producing virus-like particle for vaccine, so animal vaccine as well as human vaccine. So this particular virus-like particle has three different structural proteins. It's produced inside insect cells, and what you're hoping for is the whole cells will assemble the virus-like particle and give you the particle as you want. So we do the analysis, like we look at it under TEM, we checked it under DLS and checked that the size is correct. We also use a method what we call asymmetric field flow fractionator, which actually allows us to fraction out and check the different fractions and peaks, what they are. So for example, we get some aggregates, it tells us the size of the aggregates. This is our VLP peak, so it tells us what's the size. You have some misformed one. But what's really important is it does tell us that in that particular manufacturing condition, some proteins do not assemble into VLPs. So they have to improve in their purification methods to make sure that they remove the soluble proteins, the non-assembled one, before they take it into any trials. So that's one thing. So really, that gives you in terms of what we do in the facility, what are the capabilities and what are, some of the things we can do to apply to your project. And most of the time we work very closely, with the research group. Like we sit down, talk to you, look at, what's the best strategy for you in terms of cost, in terms of time, and in terms of, what you want to use it for. So you can say, I say all that, do I have a good track record? So we've been at UQ for more than 10 years and we produce this whole different range of different proteins. So feel free to come to talk to us if you're interested in proteins. And also that shows you the different groups that we've worked with in Australia. So I'm still trying to find someone in Northern Territory to work with. That's the only one that I didn't have. So everywhere else is pretty well covered. And in terms of, say, overall pictures, so in Australia, I was saying that, we've worked with 16 universities, different government research organizations, 12 companies, and we also have international academic and industry contracts that we work with. So the UQ Protein Expression Facility actually was invited to join the European Network for Protein Facilities, so they call it P for EU, and we are actually one of the non-EU participating facility. So most of the facilities are all located in the EU. So this is actually really useful because we share protocols, we do a lot of testing together, we share data, so it allows us to actually have access to the latest protocol or the latest technology, and we even share materials. So if you like to work with us, there's reason why you can consider us, because of our track record, our expertise, our capabilities, and our platform. And I'm proud to say that, the team actually won the Chancellor Award for Team Excellence in 2013 as a recognition for their long-term commitment and contribution to UQ. So once again, to sum up, this is our platform. And like I say, you can enter at any point. You can leave at any point. It's entirely up to you. Just come and have a conversation with us, what you think is the best and what we can do for you. And then, to find out more information, so you can go onto our website, which is basically UQ website /PEF. Or if you type, Protein Expression Facility, you come on Google, it'll come up. One thing I want to highlight to you is we have a resource center, so you can log in actually using your UQ staff or student password. So what it does is it allows you to get access into our reagents and materials that could be useful for you. So I was saying, like, how some students, they just go to the lab next door, pick out a vector, try to clone it, and then they have problems. We have seen this before where they pick out a tube with, say, 2 different vectors in it, and then they go, oops, that's why my cloning didn't work. Or they pick out a strain that is not the strains that they actually want. But here, at least, what I can guarantee is with QC, all our vectors, our reagents, and you can go in and actually purchase them at very, very low cost. Do you remember all these are heavily subsidized by UQ? For UQ users only. So you can log in and get the vectors that are interested, the cell line or cell whole strains, or say even some materials for purification or for cloning that you need to have. You can do that. So with that, I just thank you for your attention, and I'm happy to take questions.